This is going to be about new drugs under development as neuroprotection. Now, what do I mean by neuroprotection? Here's what I mean. I hear some sort of and this is not projecting. Okay, let's try it with the cursor. All right, that's pretty good. Neuroprotection means some kind of treatment that actually addresses the underlying disease process rather than only addressing the symptoms, the superficial symptoms. It's nice to address the superficial symptoms, but it would be better to stop the progression of the underlying disease process. And of course, if you want a real cure, then you'll reverse the underlying disease process so as to regain the things that have been lost, the symptoms that have been lost. That is well in the future for this disease. So what do we mean by neuroprotection right now? Well, if this is years down here, and over here is grade of disease by some random arbitrary scale, you have the person getting slowly worse over the first couple of years. And the red line shows what would happen to them if we did nothing. They would just continue to get worse at the same rate. If they have a symptomatic treatment, then they suddenly get better when the symptomatic treatment is started, and then they will continue to get worse at the same rate that they would have gotten worse had they not done anything, except that at any given point in time, there will be a difference between where they are on the symptomatic treatment and where they would have been. And this assumes that the symptomatic treatment is continued, not just a one-shot thing, but continued on a regular daily or whatever basis. Now, neuroprotective treatment, that's where the person has been getting worse as before, and now you start the neuroprotective treatment, and now the rate of worsening has been lessened. The slope is a flatter slope, which means that they'll get to the fully disabled state, they'll get to each of these states at a later point in time. Okay, that would be very nice. If we can slow this rate by about 30%, that would probably be good enough to merit actually marketing the drug and approval by the FDA and marketing the drug. Now here's a list of the drugs that are in development by pharmaceutical companies for PSP or Alzheimer's. In other words, these are tau-related drugs. Remember this morning I told you about how tau is the protein that seems to be at the root of the problem? Well, drugs that address the tau problem that's the main area that is being addressed by drug companies and by other researchers. So I showed in red here on the right the PSP trials. There are two of them that are presently underway. One is from AbbVie. The other one is from Biogen down here. I'll talk about those in a little more detail in the subsequent slides. Those are both tau antibodies. There's another tau antibody that is being developed by UCB. It's a different tau antibody, and that will start trials in PSP sometime within the next year, maybe 6 months if we're lucky. And then there is this stuff. This is, I defy you to pronounce this. The way this is pronounced is O-glycnicase. O-glycnicase. Well, what is O-glycnicase? The tau molecule, like most proteins, isn't just its sequence of amino acids. It also has little chemicals that are attached to it at different points. The most common such little chemical to be attached to a protein is a phosphate group. And the phosphate in PSP, in fact, there's abnormal phosphorylation of the tau protein. And this may be what makes the tau molecule misbehave in PSP. We don't know if that's the ultimate cause or if something else is causing the abnormal phosphorylation. So too much phosphorylation causes the tau to misfold and aggregate. Okay? Instead of being a nice spaghetti in boiling water, instead it's spaghetti after the water is boiled away and it sticks to itself. So that is enhanced by excessive phosphate on the tau. But there's another little molecule, if attached to the tau. It's a type of a sugar. It's called N-acetylglucosamine. And that sugar actually does the opposite. That sugar inhibits the misfolding and aggregation of the tau. And there's an enzyme that attaches, it's a protein, an enzyme, that attaches the sugar to the tau. And if you can inhibit that enzyme with a drug, then you can reduce the propensity of tau to misfold and aggregate. Most drugs actually work in that fashion of inhibiting the action of some enzyme. Now, these other things, this neurotrophic inducer, neurotrophins are a class of proteins, small proteins, that cause brain cells to grow or to repair themselves. So inducing them, and it's under very delicate control. You don't want too many brain cells to grow too much because that's called cancer. But you do want the brain to be able to maintain itself. So inducing neurotrophins is another approach to the tauopathies. This disease here stands for non-fluent variant primary progressive aphasia that I explained this morning. That's a type of speech problem, which is not a slurring, it's a difficulty thinking of the right word. Is this where we're supposed to be? Yes. Okay, now, so tau antibodies. You can give antibodies, or you can make the patient develop antibodies in two ways. One is by infusing the antibodies directly, and the other is by giving the person a protein or a carbohydrate or something that will induce their own immune system to create antibodies. That's how most vaccines work. The flu vaccine, for example, takes proteins that are in the flu virus, or the whole flu virus that's been deactivated, injects it, and the person's immune system creates antibodies directed against that entity. So, if you want to produce antibodies against the tau protein, one way of doing it is to just create antibodies in a factory and infuse those that would have to be intravenously into the person. And how do you make antibodies in a factory? Well, you program bacteria, usually, to create those antibodies by giving them a human gene that does it, or sometimes you can do it with stem cells or some other cells. So antibodies are proteins of a specific, proteins of a specific structure that recognizes and destroys another specific protein or other molecule with the help of the immune system. And if you infuse the antibodies, it does not make the person's immune system make more antibodies. It's just whatever you get in that dose is what you have to use. And typically, it'll last for about a month. And so the infusions of antibodies has to be repeated every month. And as I said, it has to be intravenous. It takes an hour or more to give the adequate number of antibodies. And this is what a rendering of what antibodies look like. These are protein strands. The vaccine is a protein resembling a target protein in a safe form that's recognized by the immune system, which destroys it and also destroys the target protein. And this, depending on the vaccine, may last the rest of the person's life. Like, well, a flu vaccine lasts only a few weeks, a couple of months, but a polio vaccine lasts your whole life. And other things last somewhere in between. And this can be usually by intramuscular injection, in other words, a shot in the arm, in the rear end, or sometimes it's given orally or intravenously, depending on the vaccine, depending on how it gets absorbed into the system. But usually it's an intramuscular injection. Now, decades ago, there was an attempt to treat certain diseases with, certain brain diseases with vaccine, with a, in other words, active immunotherapy where they inject a protein that induces the immune system to react. They tried doing that for a brain disease. And the result was that the person developed encephalitis. Their brain just became very inflamed. It was the body's immune system overreacting to this protein and just attacking brain cells. And that put a damper on all immunotherapies for brain diseases for decades. And it was only like in the past 5 years or so that this has been revived, mostly in the form of passive antibodies. That's this first column. But now that we've seen that these passive antibodies are quite safe to use, now a couple of drug companies are starting to slowly, carefully get back into the business of developing vaccines for brain diseases. Now, here's our cemetery. Here's our list of neuroprotective treatments that have been proposed and tried in proper, large, expensive trials. I'm not talking about one neurologist giving it to a half a dozen patients in his practice. I mean major, hundreds of patients in some of these trials. The Aricept actually would not be neuroprotection. That would just be symptomatic. That does not work for PSP. Maybe for CBD, but not for PSP. Azilect, there have been trials in Parkinson's where it's kind of equivocal, but in PSP it does not provide neuroprotection. I don't think I need to go into the details of all of these, although lithium, very, very old familiar drug used for bipolar disorder or mania, it turns out it has an interesting action on the tau molecule. It prevents excessive phosphate attachment. Remember I told you that phosphate groups will increase the propensity of tau to misfold and stick to itself. Well, lithium interferes with that. So it's been tried in PSP, but it turned out it caused too many side effects, and so it didn't get to the point that we were able to tell if it was actually working. Methylene blue is another very old drug. It's approved, it's on the market, it's used in certain medical conditions. It turns out it does not work, at least in Alzheimer's. It hasn't been tested in PSP. That was going to be the next step. Mirapex is a drug that's on the market for Parkinson's. It's a symptomatic drug, but there's some evidence that it may have neuroprotective properties, and in fact it's been tried in PSP and it did not work. Riluzole is a drug that's on the market for ALS, amyotrophic lateral sclerosis, Lou Gehrig disease, where it does have a neuroprotective effect. It does slow the progression of the disease, but just by a little bit. It really makes a difference of only a few months. But it's universally prescribed for people with ALS just because there's nothing else. Salsalate's really interesting. This is basically aspirin with a tiny little change. It's aspirin. It's a non-steroidal anti-inflammatory that does have some effect on tau, unlike other non-steroidal anti-inflammatories. Turned out it did not work for PSP. Very sensitive system here. Tideglusib does the same thing as lithium on the phosphate. I'll talk more about Tideglusib on the next slide. That didn't work. TPI 287 is an anti-cancer drug. Interesting, why should an anti-cancer drug help PSP? I mean, in theory, why should that work? Very different diseases. Well, because in cancer, there's too much cell division. Okay, now cell division, when a cell splits into two, while it's in the process of splitting into two, the chromosomes have to line up properly. So the chromosomes will reproduce themselves, and then you get one copy of each chromosome that goes to each of the two offspring cells. And in order to get the chromosomes lined up properly, you have these kind of cables, kind of these strands inside the cell. And those strands are made of microtubules. And that's what tau does normally, is stabilize microtubules. That's the job of tau in our brains all the time. And so the thought was that if you interfere with the action of, you enhance the tau, and so you'll enhance the stabilizing effect of the microtubules, and then you'll prevent the mitosis, the cell division from taking place. Didn't work. Valproic acid has the same action as the Tideglusib and the lithium. It reduces phosphorylation of tau. Valproic acid, this may be familiar to you. You may have heard of Depakote. This is I think it came on the market in the late 1970s for epilepsy. And it's used not only for epilepsy, it works very nicely. It's also used for mood disorders in psychiatry. And young plasma. I bet you've been waiting for me to get to this one. What is this one? Young plasma. Sounds like Frankenstein. That's exactly what it is. They take plasma from young, healthy people. In other words, they take blood out, separate all the cells from the blood, leaving all the proteins there, and infuse the blood into people with PSP. It's also been tried in Alzheimer's disease. Does not work. Didn't hurt, but it does not work. All right, so I told you about the about the Tideglusib. This shows a graph in the Tideglusib trial, which was published in 2014. We gave Tideglusib to a whole bunch of people with PSP and placebo to a whole bunch of people with PSP at the same time, 2 different dosages of Tideglusib. And here's the PSP rating scale score. And you see they're basically all the same. There was no advantage of the Tideglusib over the placebo. Okay, using the PSP rating scale as the outcome measure. PSP rating scale measures ability to walk and talk and think and move your eyes and balance and all those things, swallow. However, a subset of the patients received MRIs before and after and in the middle. And the MRIs did show a slowing of the rate of shrinkage of the brain in the Tideglusib patients. So here, it's maybe hard to see. Here's placebo. You can see there's more black space. The black space is the fluid around the brain. The white and the gray are the brain tissue. And there's more black and less white and gray as time goes by over the 1 year of the treatment trial. It's a reduction of an average of 3%. This just shows a side view of the same thing. In the Tideglusib patient, there was less of a shrinkage. It was only 1% over the 12 months. And here it shows the different brain regions to show you which areas were most responsive. Now, this may seem like, hey, this is great, but we can't treat the MRI. We have to treat the patient. And the patients did not respond using the best available measure. And there's been some quibbling about whether the MRIs were done properly, but the real decision was made by the drug company that was developing. This was a small drug company in Spain. They decided, all right, we're not going to pursue this anymore. They just quashed the whole project, and then they went out of business. So no more Tideglusib. However, the principle of Tideglusib, which is to reduce the attachment of phosphate to the tau, that lives on. That may still have potential. All right. Now, current and imminent clinical trials targeting tau in PSP. Okay, so I mentioned the AbbVie and Biogen studies. They are both monoclonal antibodies. Monoclonal, what does that mean? That means that, you see, when you give a vaccine, the immune system creates all different kinds of antibodies that are all directed against that vaccine. But they're different antibodies because they come from different bone marrow cells. So in the details, the antibodies will differ. So that's multiple clones of cells that make the antibodies. The monoclonal antibody is just one clone in the laboratory, so it's just one flavor of antibody. The AbbVie drug still has a inscrutable code. The Biogen drug, I don't know if it's been a public announcement yet, but they told me 2 weeks ago that the generic name is gozaranimab. MAB means monoclonal antibody. So the Biogen results will be late 2019, early 2020. You know, I reversed this. The pivotal results in the AbbVie will be later than the pivotal results in the Biogen. The Biogen will be late 2019. The AbbVie is about 6 months later, so it'll be in early 2020. Sorry about that. This ALS Protect, this has not been started yet. Don't know when it will start. The O-glycanase inhibitor from Acineuron, and Merck also has an O-glycanase inhibitor, a different one. They will start late this year or early next year. And the monoclonal antibody from UCB will start maybe late 2019, early 2020. So this is very exciting times. I mean, put yourself in my shoes. I've been working on PSP for decades now, and we've had zilch to offer in terms of neuroprotection. And now we have these real possibilities. Let's hope they don't go the way that sad list goes. Let's just hope. Okay, there's a little correction here. No, that's right. No, I need to make a little correction in this. Again, I reversed the Biogen and the AbbVie. The duration of the infusions is 1 hour for the Biogen drug and 3 hours for the AbbVie drug. It doesn't make much of a difference. It's a big undertaking anyway. So both of these antibodies are directed against an N-terminal fragment. What does that mean? That means it's near the starting end of the protein. Proteins have a beginning and an ending based on how they come out of the machinery in the cell. It's just 6 amino acids. The antibody is directed against just this little stretch of 6 amino acids near one end of a protein that has 441 amino acids. You remember the amino acids from high school biology? Are 20 amino acids in human proteins, arranged like different colored beads in a pop bead chain. And the order of the protein determines the function. The order of the amino acids determines the function of the protein, except where we might attach little things like sugars and phosphates. So both studies have 300 or 400 patients divided into active drug and placebo, the majority getting active drug. And the antibody is given as a monthly infusion for 1 year. I did make this correction on a subsequent edition of these slides, but then I wasn't able to upload them to the system, sorry. And after the 1 year of the double-blind phase, there's an open-label active drug add-on. And that's already in progress because a lot of the patients in both of these studies have already completed their 1 year of the double blind. The last one in the Biogen study will finish in September, and the last one in the AbbVie study will finish something like 6 months later. So that's the status of those. These kind of nicknames are given to drug trials. Used to be the nickname had to be some kind of acronym that made sense. Not so much anymore, although the letters PSP do appear somewhere in the Biogen name. So here's the timelines. I mentioned those already. So are these things likely to work? That's the big question. In fact, I'm a consultant for both of those companies, and a couple of weeks ago, the Biogen top brass summoned me to their headquarters, and we all sat around a table and they said, "Is our drug likely to work?" I said, "Well, the other drugs haven't, but I have high hopes for your drug." So why is there some reason for caution here? 3 reasons for caution. 1, the antibodies, the target that they were designed to address is not quite the type of tau that's in PSP. The antibodies target tau taken from stem cells from patients with a genetic form of Alzheimer's disease, not PSP. You may know that patients with young onset Alzheimer's, they tend to have a genetic form. It's caused by one gene, one mutation in one gene. Tends to run in families very strongly. So they took stem cells from those patients, probably from just a punch biopsy of the skin, very easy to do. They grew up some stem cells from the layer of skin that hasn't really turned into skin cells yet. And they programmed them to, they created antibodies from the tau that was in those cells. So that's not PSP tau. Problem number 2, when you develop a drug, you have to test it in animals. It's called a model. The best model we have for PSP is a mouse in which there has been genetically inserted a human tau gene, and the human tau gene is a defective human tau gene. It has a single mutation in it, which is known to cause a human disease, frontotemporal dementia. There's certain types of frontotemporal dementia that are hereditary, that are caused by a single mutation in the tau gene, and that was put into mice. The mice grew up and developed a tauopathy. They developed a neurodegenerative disease, which kind of sort of looks a little like a human tauopathy. And the humans that the gene was taken from, well, they have a real human tauopathy, but it's not PSP. It's frontotemporal dementia. And even in the brain, if you look at the brain under the microscope, it looks different than PSP. So the mouse model that we have, the best model that we have, is still not that close to human PSP. And no one has been able to figure out a better model. A few years, I don't know how many years, maybe 10 years ago, 12 years ago, CurePSP gave a very large grant, one of the largest in our history, to a very well-reputed scientist, very accomplished, productive scientist, to develop a rat model of PSP. And he failed to do it. He couldn't figure out how to do it. That tells you how difficult this is. And finally, many previous trials of neuroprotective treatment in neurodegenerative diseases have failed despite good results in the best available animal model. The trials would never have proceeded to humans if they didn't get really good results in the animal model. All right, now, finally, if, fingers crossed, if it turns out that one of these monoclonal antibodies works, after the company announces that, it would be like another year and a half until the FDA would approve it. It's not at all certain that the FDA would approve it even if the company thinks it works, doesn't mean the FDA wants to approve it. So there's that little bump in the road. But let's say the FDA does approve it. It may be a year and a half before they would actually be able to get the drug on the market. So over that period, they are allowed to offer the drug for free to qualifying people under certain very restricted circumstances with a lot of oversight and monitoring. It's called an early access program. So the drug would be free. The cost of the administration, this is another edit that I made that couldn't be uploaded, would be free to the patient and the family. So the infusion center, where you have to go and sit in a chair for an hour or 3, there's a fee for that, and for the staff. So, but that would somehow be paid for. They don't yet know how, but it would somehow be paid for. The details of who would qualify and what kind of testing along the way would be required, those details are still up in the air. But it would not be as onerous as being in the actual trial. So that's where we stand right now. And I'd be happy to take questions. Hang on, the mic's coming.
How do people get chosen for the trial?
How do people get chosen for trials?
Yes.
Well, first of all, you understand that the Biogen and the AbbVie trials have finished enrolling people. The UCB trial and the other ones, yes, they will be starting to enroll at some point. So the answer to your question of how do you get into a trial? It really helps to be an existing patient of a center, a neurologist, typically at an academic, at a medical school where the trial is taking place. These trials they'll have maybe a couple of dozen places in North America and a few in other countries. So, and these are the top PSP centers in the country. Now, sometimes such a center will include a person in a trial who was not already one of their patients if they get contacted by that patient, and they will bring the patient in for a screening, and that works as well. But it may be a little more difficult to get in that way because the centers will naturally want to favor the patients that they already know. You know, being in a trial is a very difficult thing for a patient and a caregiver. It's a lot of traveling, a lot of sacrifice on their part, for which we are extremely grateful. But it also means that the staff, the coordinator and the neurologist running the study want to be really sure that the people they enroll in the study are going to be robust. They're going to be easy to deal with. You know, they won't decide to quit halfway because they think the drug isn't working. They want a good study patient. And if they've only met you once, they maybe can't be quite so confident of that. But if they already know you on an ongoing basis, it makes it easier for them to be confident of that. Good question.
Do the trials, are they safe? Okay, darling. Are the trials safe, sir?
So far, the AbbVie trial, the Biogen trial has announced its safety results, and it is very safe. You could barely distinguish the side effects from the placebo group. The AbbVie has not yet announced its safety results, but I expect it'll be about the same.
What about the one that's going on, sir?
Those are the AbbVie, that's the AbbVie and the Biogen.
That's the AbbVie and the Biogen. I thought there was a new one, PSP drug, that was
There's a whole bunch of new ones that'll be starting in the next year.
Yes.
So we don't know how safe they are. They haven't been tried yet in people with PSP at all.
That's right.
Right?
All right, thank you.
You're welcome.
So both the clinical trials are in phase 2 or they are
Phase 2.
Phase 2, okay.
But there may not need to be a phase 3. If the FDA, with orphan drugs like this, if the FDA is happy with the phase 2 drug, if it's convincing enough, they'll approve it without a phase 3.
Okay, so the Biogen is more recent now?
The Biogen started earlier.
Right.
Yes.
Right, so that will be out in the market sooner than the,
Well,
if depending
the
on.
results will be available
Right,
sooner.
yeah,
You
yeah.
never know how long it takes the company to ramp up the production and get the
Right.
drug on the market.
And what will be the difference between the two drugs?
They both target, they're a different method of producing the antibody, but
Right.
the two antibodies target the same section of the tau
Tau.
molecule.
Okay, okay. So they won't act as vaccine, they will be antibody medicines
Yes, they're passive, so they will have to be repeated every month.
every month, okay.
Yes,
Any idea, like every month is how many sessions? No idea, right?
one session a month.
No, how many? With the length of the infusions?
Oh, one of them
A
is 1 hour.
year or a, no?
Oh, for as long as you live.
Forever? Okay.
Yeah, lifelong.
I see. Yeah, and we don't know the side effects also, right?
So far, the side effects are basically nil in the Biogen. The antibody, the AbbVie, I expect it to be similar, although we don't have data yet.
I see, thank you.
This morning, you or someone else said that if you're a person with this disease today, you're much better off than if it were 10 years ago. Is that actually true?
It's true that the disease is getting diagnosed earlier in the course than was the case 10 or 20 years ago. So in theory, if you're diagnosed earlier, then you're better off because you won't be shopping around to different doctors, getting unnecessary tests, unnecessary ineffective treatment. So in that sense, you're better off. That's the only way that you're better off.
These drug therapies.
Well, naturally, the further in the future you are when you get the disease, the more likely there is to be an effective drug, sure.
I would add to that too, that the other benefit is that there are others involved in the process, like occupational therapists and physical therapists, who are more familiar now with these kinds of things and have more germane ideas that they can help you cope with and adapt to the disease.
In case you in the back couldn't hear that, what Mr. Robinson just said is true, that other professionals, including neurologists, are much more familiar with PSP than was the case 10 years ago. Thanks in no small part to CurePSP. I have to just put in that little plug there. And if you can get a physical therapist who knows about PSP, you're better off than a physical therapist who doesn't know about PSP.
Okay. My question is, I want to understand, is access to the early access programs how different is that from access to a trial?
Good question. I should have mentioned that. That will be much more democratic. It'll be local neurologists would probably be able to do that. You probably would not have to go to a major center for that. Is that basically what you were asking?
Yes.
Yeah. But the details haven't been finalized yet.
So from a practical point of view, assuming that Biogen or whatever they progress, with the limited population of the disease, the economics dictate that the cost would be fundamentally, for the average person, impossible.
Oh yeah, the rack rate, the retail cost of the drug oh, it's going to be astronomical. But insurance companies are paying it for other drugs, other monoclonal antibodies.
How long is the process take to get an insurance company? And if assuming the approval, how long would it take for an insurance company to recognize that they want to pay?
I don't know how long it's taken in the past, but I can tell you that at a meeting I once was at a few years ago, in which there was a representative of one of the big insurance companies, we asked him exactly that question, and he said, yeah, we'll pay for it. One reason being that PSP and CBD are relatively rare diseases. It's not Alzheimer's. Another reason, unfortunately, being that even if these antibodies work at our maximum hopeful expectations, this is still, it's not a 50-year deal like it would be with a child with a disease. I think we're going to have to cut it off now, but I'll be around. Thank you all for listening and for your excellent questions.
Thank you, Larry.